unraveling the enigma of aging, an evolutionary perspective. Are you a researcher captivated by the complexities of aging? Delve into the evolutionary theories that illuminate why we age, offering profound insights into the genetic and molecular mechanisms driving senescence. Aging, characterized by a progressive decline in function, reduced fertility, and increased mortality, presents an evolutionary paradox. This seemingly detrimental trait raises fundamental questions about its origins and persistence. Why would natural selection favor a process that diminishes survival and reproductive success? This article explores the evolutionary theories that attempt to answer this intriguing question, providing a framework for understanding the biological underpinnings of aging. Early explanations propose that aging evolved to limit population size or accelerate generational turnover, facilitating adaptation to changing environments. However, this group selection argument faces a critical flaw. In most species, mortality in the wild is primarily driven by extrinsic factors like predation, infection, and starvation, rather than senescence. Wild animals rarely live long enough to experience significant age-related decline, limiting the direct influence of natural selection on the aging process. The rarity of aged animals in the wild provides a crucial clue to understanding the evolution of aging. Extrinsic mortality weakens the force of natural selection with increasing age, creating a selection shadow. This shadow allows the accumulation of late-acting, deleterious mutations in the germline as selection is too weak to effectively eliminate them. The mutation accumulation theory suggests that aging arises from the gradual buildup of these unselected, harmful alleles, leading to a heterogeneous distribution of aging-related genes within a population. The theory of antagonistic pleiotropy offers another perspective, proposing that genes with beneficial effects early in life can be favored by selection even if they have detrimental effects later on. A small early advantage in survival or reproduction can outweigh a significant late-life disadvantage, such as senescence or death. This introduces the concept of a life history trade-off, where resources are allocated between different functions, leading to a compromise between early fitness and late-life health. The disposable soma theory focuses on the optimal allocation of metabolic resources between somatic maintenance and reproduction. It argues that effective somatic maintenance is only necessary to keep an organism in good condition for as long as it has a reasonable chance of survival in the wild. Since resources are limited, organisms benefit from investing in immediate survival and reproduction, rather than in costly repair mechanisms that would only benefit a small fraction of the population that survives to old age. These three theories, mutation accumulation, antagonistic pleiotropy, and disposable soma, provide complementary explanations for the evolution of aging. They also address the question of why different species have different lifespans. The level of extrinsic mortality is predicted to be the primary determinant of longevity. High extrinsic mortality leads to short lifespans, early accumulation of deleterious gene effects, and limited investment in somatic maintenance. Conversely, low extrinsic mortality favors delayed deleterious gene effects and greater investment in a durable soma. Evolutionary theories implicitly assume an age-structured population, where individuals can be segregated by age. This predicts that aging should not arise in populations where age classes cannot be assigned, such as bacterial populations. In multicellular organisms, aging generally requires a clear separation between germline and soma. Studies on organisms with different reproductive strategies, such as oligochaetes and hydra, support these predictions. Artificial selection experiments, particularly in Drosophila melanogaster, have provided strong support for evolutionary theories of aging. By restricting reproduction to later ages, researchers have consistently extended the lifespan of selected populations. These long-lived flies often exhibit reduced fecundity, supporting the idea of a trade-off between fertility and survival. Similar trade-offs have been observed in nematodes, although the results can vary depending on environmental conditions. Comparative studies across different species offer further evidence for the role of extrinsic mortality in shaping aging rates. Adaptations that reduce extrinsic mortality, such as wings, protective shells, and large brains, are generally associated with increased longevity. Field observations comparing opossum populations with different levels of predation have also found that lower extrinsic mortality is associated with slower aging. At the molecular and cellular levels, the disposable soma theory predicts that the effort devoted to cellular maintenance and repair processes will vary directly with longevity. Studies have shown that long-lived species exhibit lower levels of oxidative damage, higher concentrations of antioxidant enzymes, and greater DNA repair capacity compared to short-lived species. Furthermore, cells from long-lived species exhibit superior stress resistance, supporting the idea that effective somatic maintenance and repair govern the rate of aging. Many organisms live in highly variable environments, leading to the evolution of plasticity in life history traits. Environmental conditions, such as temperature and food availability, can influence longevity. In some cases, periods of famine can trigger metabolic switches that extend lifespan without sacrificing subsequent reproduction. The nematode C. elegans provides a well-studied example of this type of plasticity, with the formation of the dower larva under conditions of high larval density and low food availability. The phenomenon of caloric restriction, where reduced calorie intake slows aging in laboratory rodents, may have parallels with the invertebrate response to resource scarcity. Caloric restriction is associated with enhanced stress resistance and a partial interruption of fertility, suggesting an adaptive physiological state to wait out periods of food shortage. The most pronounced impact of environment on aging rate is illustrated by the differences in longevity between queens and workers in eusocial insects. Queens, living in protected nests, can live for years, while workers, exposed to the dangers of foraging, typically live for only weeks. This dramatic difference in lifespan is mediated by pheromone exposure and the rate and composition of food supplied to the larvae, highlighting the profound influence of environment on aging. While most discussions of aging focus on mortality, the reproductive schedule is equally important in terms of fitness. Many aspects of reproductive senescence can be explained in the same general terms as physiological senescence. However, there are intriguing evolutionary questions about the links between reproduction and aging, including the significance of post-reproductive survival and the effects of damage and selection on the germline. The human menopause, where fertility in females comes to a relatively abrupt halt around the age of 45 to 50 years, presents an evolutionary puzzle. While the proximate cause is oocyte depletion, the question remains why natural selection has not produced a longer-lasting store of oocytes. Several hypotheses have been proposed, including the increased risks of childbirth at older ages, the benefits of grandmothering, and the link between menopause and the evolution of human longevity and sociality. The germline must be fundamentally immortal to prevent the accumulation of damage across generations. However, individual germ cells do accumulate faults, as evidenced by the increasing frequency of chromosomal abnormalities in newborn children with increasing maternal and paternal age. Mechanisms such as special maintenance and repair systems, selection at the cell or embryo level, and the stringent bottleneck in the size of the cellular mitochondrial population in early embryogenesis may serve to screen out most faults and maintain germline integrity. Understanding the evolutionary forces that have shaped our genetic makeup provides valuable insights for investigating the molecular and cellular basis of aging. Evolutionary theories predict that multiple genes influence the aging process, including genes controlling somatic maintenance and repair, late-acting deleterious genes, and pleiotropic genes with trade-offs between early benefits and late-life harms. An important corollary of the prediction that key genes regulating aging control somatic maintenance and repair is that stochastic chance plays a significant role in the aging process. individual cells within tissues experience different random accumulations of faults, and there may be important stochastic variations in developmental processes. This means that genetically identical individuals, maintained in uniform environments, can still exhibit considerable variation in the senescent phenotype. The evolutionary theories of aging provide a powerful framework for understanding why we age and offer valuable insights for future research. By focusing on the genetic and molecular mechanisms underlying somatic maintenance and repair, researchers can identify potential targets for interventions that promote healthy aging and extend lifespan. Are you ready to contribute to this exciting field? Here's how you can get involved. Deepen your knowledge, explore the cited research and related literature to gain a comprehensive understanding of the evolutionary theories of aging. Design innovative experiments. Develop research projects that test specific predictions of the evolutionary theories using diverse model organisms and experimental approaches. Collaborate with experts. Connect with researchers in gerontology, genetics, and evolutionary biology to foster interdisciplinary collaborations and accelerate discoveries. Share your findings, publish your research in peer-reviewed journals, and present your work at scientific conferences to disseminate knowledge and inspire further investigation. Together, we can unravel the mysteries of aging and pave the way for a healthier and longer future for all.